All right, sorry for waiting. Let's start today's seminar. Good afternoon, everyone. Welcome to the Student Census RIG Seminar series for this semester. My name is Lee, and I'll be the chair of today's talk, and also one of the members in the Student-led Census Interest Research Group. The primary aim of our group is to share research presentations in the areas of sensors and sensing technologies. We hope the student seminar series will enable knowledge share and foster collaborations. Please visit our website to explore more about our organization. If you would like to present your research or interested in joining our group, please do not hesitate to email us. The title of today's talk is Cross-reactive Arrays from the Anslyn Lab for Differential Sensing. We're very excited to have Dr. Hazel Fragher here today. Dr. Hazel Fragher received her bachelor's in chemistry from Worcester Polytechnic Institute, and she received her PhD in physical organic chemistry from the University of Oregon. She's now the postdoctoral researcher in the Anslyn labs, where she studies information storage and retrieval in sequence-defined algorithms. Before the talk begins, I would like to remind you that you can always ask questions at any time by using the chat box. We'll collect the questions and ask the presenter at the end of the talk. Now I'd like to give the floor to Dr. Fragher, and the time is yours.
Great, thanks so much for that introduction, and thank you, Song Lee, for inviting me to give this talk. Let me just share my screen. Okay, so my talk today is going to be in cross-reactive arrays from the Anslyn lab. And before I start this talk, I just want to preface this with that none of the work that I present today is going to be work that I've worked on. Instead, I'm highlighting work that has been done before my time in the Anslyn lab. And I'm kind of here on behalf of Eric Anslyn. And some of the slides that I'm going to be presenting to today are also from Eric Anslyn. So just to give credit where that's due. Okay, so I want to start by talking about molecular probes. So these are just molecules or a small cluster of molecules which produce a measurable response upon binding, reacting or binding with a given analyte. And often that measurable response is going to be a colorimetric response, a fluorescence response, or some sort of electrical signal that you can read. So that's great, but how do we go about designing these probes? So the very traditional approach of making a molecular probe is a lock and key approach. And so this is where you kind of design your molecule to be highly selective and highly specific for your desired analyte. Sorry, let me make this a laser pointer. Can you all see the laser pointer? Great, thank you. So if we look at this cartoon, we could imagine that this gray box represents our highly designed molecular probe, and the only analyte that could possibly fit in this molecular probe is this orange-looking substrate. And upon binding, we get a colorimetric response where we see an orange readout. And this is a really common approach that's used in a lot of antibody and antigen tests. So for example, the pregnancy test is one of the most well-known examples for this. When you become pregnant, your body produces an HCG pregnancy hormone, and nature has already provided us a perfect or near perfect, highly selective molecular probe in the HCG antibody. And this binds really specifically and sensitively to the pregnancy hormone. So if you have them in solution, you'll make this host-guest complex where the antibody has bound to the pregnancy hormone. And then if you have like a solid substrate that has more of an HCG antibody on the solid substrate, you can then capture and immobilize your pregnancy hormone on a solid substrate. And oftentimes in the at-home pregnancy test, these antibodies will be labeled with a dye, and in that way you can clearly see a colorimetric response if you have this pregnancy hormone. And so this is a really powerful technique, but this is so, but unfortunately for us, nature hasn't provided antibodies for every single molecule that we could want to detect. So oftentimes you have to go and design and build your own small molecule probe, which can be really challenging. And overcoming this cross-reactivity with interferences is really challenging. And as a result, the development of useful and highly specific small molecule probes has historically been pretty slow. So in the Anslyn lab, we take another approach, and this approach has been inspired by nature. So the human nose can detect up to 1 trillion scents, and it's estimated that the human tongue can taste up to 100,000 different flavors. And so in really basic, I'm not a biologist, so in really basic terms, how this happens, our noses have scent receptors, our tongues have taste receptors. And when a molecule that we taste or smell interacts with that taste receptor, the receptor sends cellular signals to our brains. However, when we drink a cup of coffee, we don't have, a receptor that's highly specific for coffee. If we're drinking a glass of wine, we don't have a receptor that's highly specific for wine. Instead, we have receptors all over our noses, all over our tongues, and they will kind of react and bind with all of the different tastants that are in wine. And they'll react a little bit differently with all of the molecules in there, and then send all of those cellular signals to our brain, so that our brain does a little bit of pattern recognition and then tells us, oh yes, you've just drank a whole bottle of wine. So we want to kind of copy that approach, except with small molecule sensors. So our approach, or the approach in the Anslyn lab, is to make an array of cross-reactive receptors. So now we're not trying to make probes that are highly specific for an analyte. We're just trying to make, a probe that will bind an analyte and then make a whole array of that. And now it does not have to be specific for that analyte, because when we add the analyte, we'll see a different response across the whole array, our whole sensor array. And then we use machine learning techniques or statistical techniques to ask, this algorithm, can you identify a fingerprint for our analyte? And then, that statistical tool or that algorithm can tell us, oh yes, this is the analyte that you've added to this array, or this is the complex mixture that you've added to the array. So now the array does not have to be specific for an analyte, because if you added a different analyte that the cross-reactive array still reacts with, you should in theory get a different fingerprint, and you should be able to identify that different analyte. So this work has really inspired a lot of the projects that are done in the Freshman Research Initiative Super Sensors track. And so the work that I'm going to be talking about today has all been done by the FRI freshmen and published by them. The kind of design idea behind the projects done in the FRI Super Sensors track is to find synthetic receptors that will bind to an analyte of interest, and then take those receptors and add diversity to them in hoping that they can make a sensor array for that analyte. And one of the major targets in the past for the FRI Research Lab has been constructing an array of peptide-based sensors to fingerprint wine varietals. One way of determining what kind of wine you're drinking is to be like this guy and use your sense of taste and your sense of smell to drink the wine, and then figure out, what varietal am I drinking? And so this project that I'm about to talk about, all the projects I'm about to talk about that involve wine have been in collaboration with Professor Hildegard Heyman. She is a professor at UC Davis, and she's a sensory scientist, so she's a really well-known wine taster. So, we could distinguish between different wines by using our sense of taste and smell, or we could not drink the wine and create a peptide-based sensor array. So in this peptide-based sensor array, we would want to see a unique response, whether we've added Zinfandel, Cabernet Sauvignon, Pinot Noir, Shiraz, or any other type of wine. And we should be able to see a unique fingerprint response to that peptide sensor array. So other than our boss's very obvious love for wine, why are we interested in trying to fingerprint wine? One of the reasons is that wine is a really complex solution. And if we want to show that sensor arrays are useful, perhaps for, medical diagnostics or stuff like that, we would want to show that peptide or these cross-reactive arrays can work in a complex solution. Also, wine has a large amount of these tannins or flavonoids molecules in them. All of these flavonoids are structurally pretty similar, so they would likely all react with a sensor in a similar way. And the composition and structure of these flavonoids are very dependent upon the biochemistry of the grape. So this might change depending on the type of grape you use, depending upon when you harvest the grape to make the wine, what kind of soil it's grown in. All of these things can influence what type of flavonoids you get in that wine. And finally, not all of these flavonoids have been characterized in wines. So, if you are able to make a peptide array that can distinguish based on the flavonoid in the wine, you're really distinguishing based on molecules that you don't even know whether or not they're in there. So it's kind of like a very robust way of distinguishing between wines. So experimental design for the sensors that go into the sensor array is based off something called an indicator displacement assay that has been developed in the Anslyn lab. So to make the probes or the sensors that go into these sensor arrays, you have kind of a ternary complex where you have a copper species that coordinates with a peptide chain and also with a dye. So in this case, pyrocatechol violet. Upon addition of a flavonoid, the flavonoid should displace the dye, as you can see over here, and instead bind to the copper. And when you displace the pyrocatechol violet dye, you should see a color change. So in this 96 well plate, you see over here, they're showing that they start with this sensor with the peptide, copper, and pyrocatechol dye. And then as they add in, these flavonoids, you see a color change that can be measured by UV vis. And because this is done in the Freshman Research Initiative labs, there's a lot of potential for doing a lot of replicates. So they did 7 trials to make sure that what they're seeing was a response of the flavonoid in binding the copper and displacing the dye. So that's how the indicator displacement assay works. The reason why they have a peptide chain also bound to the copper is that here is where they can add all of that diversity to the sensors by just using different peptide chains. So a lot of different peptide sequences were made for this study. They're made by a whole bunch of different students and professors working in this FRI research over 2 different semesters, and all of these were characterized by ESI MS. And 9 of these peptide sequences, the ones that are highlighted in orange, were then used for the following study in the indicator displacement cross-reactive arrays. So once the students had these arrays of 9 different sensors, they then added 6 different wines, 6 different red wines to their arrays of cross-reactive receptors and looked at the response of the arrays with the different wines. So they looked at Cabernet Sauvignon, Merlot, Pinot Noir, Shiraz, Zinfandel, and Beaujolais wines. And then they took the absorbance response, or they took the response of their cross-reactive array, and then they performed a linear discriminant analysis on those responses. An LDA is a statistical tool which will take all of your observations. You tell it, oh, this observation came from Cabernet Sauvignon and this one came from Shiraz, for example. And then the statistical tool will try to find the maximum amount of variances between all of those observations. So in this LDA on the left, you can see that all of the 6 different wines are pretty distinct in the LDA, meaning they all have a distinct fingerprint. And just to say this again, all of this work was done by the freshmen in the FRI track under the guidance of Rick Anslyn and the other professors running this lab. And then if you take this 2D LDA and then go to a 3D plot, you can get even greater distinction between the 6 wines. So with this success, the FRI labs went a little bit further and they asked, if we mixed 3 different wines, could our sensor arrays figure out what composition of 3 different wines are now in this blend? So they mixed Cabernet Sauvignon, Cabernet Franc, and Merlot together, and then added that to their sensor array, looked at their responses, and then built a model that could predict the composition of those 3 wines in the blend. Down here is a table showing the root mean squared error of their prediction for the wines. A perfect model that completely 100% gets all of it right would have an RMSEP of 0. So these errors, I'm told, are a little bit high, but Eric then went and did this experiment where he mixed 3 wines together and asked the faculty to drink the wines and try to figure out what percentage of wine was in each blend. And in Eric's own words, we ran this experiment with our faculty and the device beat the pants off them. So I think the model was better than the faculty. So the Super Sensors FRI is taking it even further. They did even more studies with these wine varietals, and they were able to discriminate with an LDA, discriminate the same wine made from the same grapes, but those grapes were harvested at different stages of maturation or different stages of maturity. And they were able to see, based on the flavonoids in those wines, distinct LDA profiles. They went then, again, with the help of Professor Hildegard Heyman, they were able to correlate this UV-Vis absorbance from the indicator displacement assay to perceived astringency and bitterness in the taste of the wine. And in their paper, they say this is the first reported correlation. I have to move this bar. Okay, this is the first reported correlation of human descriptive sensory analysis of red wines and synthetic receptors. So the FRI lab has since moved on from wine, and it's now under the guidance of Diana Olivares Zamora. And she recently published a paper in JACS where they were able to use an array of SOX-based peptides to distinguish between isoforms of the MAPK kinase And the really cool thing about this paper, and I won't talk too much about it, is that they are able to detect and determine the amounts of these three different MAPK kinases in one single sample, so they're all together, in the presence of other isoforms, so a complex mixture, and from cell lysates, so an extremely complex mixture. So that brings me to the conclusion of my talk. This traditional lock and key approach to molecular probes, it's a really powerful technique, and it definitely has its place. For example, if I take an antigen test to see whether I have COVID or not, I don't want to sit in my bathroom and do, some pattern recognition. I want a yes or a no. But for things where you can take your results and put them through a statistical tool like an LDA, you can get some really powerful results from these cross-reactive arrays. And so the work that I've talked about today is only a sampling of the work that has come out of the Anslyn lab. But there are some really cool papers out there that don't come from the Anslyn lab. For example, the Susslich group created a molecular nose that could read the gases in the headspace of meat. And with this device, they were able to differentiate between 5 different meats and then go and classify the meats as fresh, less fresh, or spoiled. The Azenbacher group created a sensor array of supramolecular receptors that was able to differentiate 10 different anions in water, and water is the hardest media to sense ions in. They then took this array and went further and distinguished between a whole bunch of different toothpaste brands. I think they were looking for which brand of toothpaste had the highest fluoride concentration. Another group published a chip in Nature Communications where they functionalized the chip with different silicon groups and then looked at the electrical response and were able to differentiate between 5 different explosive molecules down to the parts per quadrillion. And finally, Rotello and co-workers used fluorescent probes to analyze blood serum from patients who are either healthy or those living with liver fibrosis. And the fluorescent probes were able to make a fingerprint that distinguished the serum as either healthy or fibrotic. And this is particularly impressive because they did not know any biomarkers for liver fibrosis. They just added it to the serum sample. And the serum sample is enough just to be a pinprick. So they're working on making this as a point of care device for the diagnosis of liver fibrosis. All right, that concludes my talk. Thank you all for coming, and I'd love to take any questions.
Thank you so much for your amazing presentation, and the topic is really interesting. Let's see. Please, you can just unmute yourself and ask question directly. I think please.
Oh, sorry.
Yeah, let's wait a few seconds for the audience to ask a question. In the meantime, I'm really curious, have you try to differentiate the same type of wine from different brands?
Yeah, in some of those papers that I presented, they took I think it's Zinfandel wine from different wineries. And then a lot of them clustered pretty close together on the LDA, but there were a few that were distinct.
Oh, okay, that's cool. And I have another question. Can you go back to the slides with the prediction of the mixture of the wine?
This one?
Yes, yeah, this one. There's a outlier in the right bottom corner.
This one?
Yeah. Is that because the training set doesn't cover the range there, or why this one doesn't make a good job to predict the correct ratio?
That's a good question. And I don't actually know the answer to that question. I think the error in the model is kind of high. It was something like so Eric was telling me if you had like a 30% composition of one wine, the model would be able to tell you that you have a 15 to 45% composition of the wine. So it's definitely rough. Yeah. So it could definitely use some improving, but I think, in the MAP kinase project where they sense 3 different MAP kinases in the cell lysate, they do get much better model results for that.
I see. Yeah. Maybe using more large training set will also improve the accuracy.
Yeah, and sometimes, if you add more sensors to your sensor array, you can kind of encompass more variations.
I see. Yeah. Okay, so if there's any other questions, if you have any questions, you can just email us. I post our email in the chat box, and you can always email us, and we'll deliver your question to Dr. Fragher, and then we'll get back to you. Okay, so I'll like to thank you again for your great presentation. I wish you all the best in your future works, and I'd like to thank our audience as well for joining us today. So our next talk is given by Hongru Ding on April 6th, 2 weeks from today. We're very much looking forward to seeing you again. Thank you.
Thank you.